Well, hello, everyone. Thanks for coming. And so it's my pleasure to introduce Max Chin, who is going to be talking to us today about our favorite fish. And so Max is coming to us from Texas A&M, where he did his undergrad, and he worked with not one, but two pretty fantastic scientists, Gil Rosenthal studying swordtails, and Keith Blackman doing some evolutionary simulations and phylogenies with mammals. And now he's joined my lab and Andrew Whitehead's lab, and also now collaborating with Graham, collaborating with everyone to unravel the genomic mysteries behind the Amazon molly, which is what I believe you're going to tell us about today.
Exactly.
Okay, take it away, Max.
Okay, perfect. Thank you so much, Kate. I will get started now. All right, as usual, I have an extremely long slide deck to kind of get through today, so I apologize if I end up speaking a little bit quickly, but hopefully you guys are able to kind of follow along. So, our presentation today is titled Reversing the Ratchet. Obviously, kind of some big aspirations there, but mostly what I'm looking into is these mechanisms, which I think are kind of restructuring and allowing for gene flow in the absence of recombination. So, we will start off super broadly by just thinking about the distribution of reproductive modes throughout the tree of life. So we can broadly categorize reproduction in many different systems into either being unisexual, right? We'll define this as any form of reproduction which does not involve gametic fusion, or sexual reproduction, right? In this case, illustrated beautifully here by these swordtails. And we'll define sexual reproduction as any form of reproduction which involves fusion of gametes. One thing we'll see is that there is a vast kind of distribution of these different reproductive modes throughout the tree of life. And something that's been studied quite intensively is what kind of guides the distribution and also the prevalence of these different competing reproductive modes as we move across different taxonomic scales. So one thing that's important to acknowledge, first of all, is that sexual reproduction definitely carries costs. We know by now about things like the cost of males, right? When we invest into two different gamete types, we basically decrease the ability at which we're able to grow our population by a factor of 2. And so this is illustrated beautifully here by this figure on the right, where we are comparing rates of population growth in our sexual kind of species up top, and then also our asexual lineage below. And you can see that rate at which we're able to grow our population is divided by 2, right? And so our asexual populations are typically going to be able to grow at faster rates and reach those large population sizes faster than our sexual populations. There's other kind of costs we can think about as well, things like the cost of meiosis, right? Whenever you're producing gametes which have some form of ploidy reduction involved in them, you are going to be diluting your kind of contribution to future generations, right? And this is almost universally true as we look across different forms of sexual reproduction. Obviously not the case if all your gametes are functionally identical to you, in the sense that they're diploid and don't have any kind of gametic ploidy reduction involved in the kind of unogenesis process. We can go on and describe all these other different costs that are associated with sex, but big takeaway is that sexual reproduction is costly, right? But at the same time, we also know that sex is pretty ubiquitous as well. This is especially the case when we look across our higher order kind of taxa, and we see that sexual reproduction is by far the norm when we look within these specific clades, which are more derived, we can say, right? So one of the reasons why we think this form of sexual reproduction might be so ubiquitous is because it carries the advantage of being able to organize your genomic variation through the process of recombination. So there's a couple of different models that have been used to describe how this recombination process allows for basically restructuring of haplotypes. What we'll be focusing on today is this idea of recombination being a mechanism that allows for increasing the efficacy of purifying selection. And this is illustrated best by, I think, by this idea of Miller's ratchet, right? Which some of you guys may have heard of before. But to illustrate this, we are going to start off with a really simple scenario where we have a distribution of haplotypes kind of within our population, right? Here, 5 haplotypes with the red stars on those haplotypes representing, we can consider deleterious mutations. As we move through the process of, as we move through generations and kind of experience varying levels of genetic drift, we're eventually going to lose some of our least mutated haplotypes simply through the process of drift, right? No selection kind of acting within this population at all. This is, of course, true whenever you're talking about finite population sizes, which are all populations because they have finite size. Once we lose that least mutated haplotype, without the mechanism of recombination, we aren't able to regenerate that least mutated haplotype, because we can't remove deleterious mutations from the background on which they arise. And so eventually what this is going to result in is that the average number of mutations that's present within our population is only going to increase in number over time, hence this kind of ratchet-like behavior, right? The number of average mutations can only move in one direction. Based off of the Muller's ratchet, we can kind of make this prediction that asexual lineages which do not practice recombination should be both rare and also evolutionarily short-lived. Which brings us to kind of a major paradox. So one part of that kind of prediction is true in the sense that asexual lineages are certainly rare. They represent tips on evolutionary trees that oftentimes don't diversify to the extent which we see closely related sexual lineages. But the other part of that hypothesis is somewhat less supported in the sense that there appear to be some of these asexual lineages which are actually quite evolutionarily ancient. How then do these asexual lineages, which are evolutionarily ancient, compensate for the cost of asexuality? And this is kind of the guiding question which a lot of my dissertation is focused on investigating. So we can start off by first thinking a little bit about these alternative mechanisms which might be able to fill the role of recombination. We'll think about three mechanisms specifically today. First of all, we'll think about the process of mitotic recombination, specifically how gene conversion can restructure haplotypes in very much the same way that we think meiotic recombination typically does. We'll also think about introgression as a source of novel gene flow into these asexual systems, which aren't able to kind of generate that novel diversity themselves. And finally, we'll think about some of the potential reasons why asexual lineages might actually have reduced paces of molecular evolution. So, and of course, we'll also think a little bit about how each of these three different compensatory mechanisms or putative compensatory mechanisms can interact with one another as well. So let's dive into each of these a little bit more. I mentioned mitotic recombination, and we'll think about that primarily in the context of gene conversion, right? Like meiotic recombination, mitotic recombination typically starts off from the place of a double-strand break, right? You have some double-strand break within your DNA sequence, which you want to try and repair if possible. Typically, this will involve some form of strand invasion and then multiple different mechanisms which can resolve that double-strand break. But one kind of unifying factor across all of those mechanisms is the potential for these kind of regions of heterochromatic DNA to be generated, which are typically repaired using the process of mismatch repair, which, if occurring in a certain direction, can actually lose heterozygosity at a locus. So these are represented by like our red boxes here, where we see that kind of heterogeneous DNA, and we can see these black boxes represent where these gene conversion events have occurred. Before our kind of double-strand break, we obviously have these two haplotypes being heterozygous across the entire kind of simulated strand here. And after this kind of mismatch repair process, you see that we've lost heterozygosity within that specific kind of like black box region, right? And so, like I said, there's lots of other different mechanisms by which we can repair these double-strand breaks, but they're all unified in their ability to generate these kinds of gene conversion events, which show up as signals of loss of heterozygosity. So, in addition to that kind of gene conversion mechanism, another kind of compensatory processes we'll think about is introgression and how that kind of provides novel targets that selection can act upon. Here we'll use the specific example of double-wave rotifers, which are some of the oldest kind of eukaryotic asexual lineages out there, and how, in this case, we have pretty strong evidence of them preferentially expressing these horizontal gene transfer-derived genes under these conditions of biotic stress. In this case, both of these plots are representing two different species of these double-wave rotifers under two different time conditions when exposed to a kind of bacterial infection. And what we see is that comparing our down and our up-regulated genes and our contribution of our horizontal gene transfer-derived genes to these two different categories as compared to those genes which are not significantly differentially expressed, we see a lot higher representation of these horizontal gene transfer-derived genes in our kind of down and up-regulated genes under this kind of stressor. And finally, that last kind of mechanism we'll discuss is this idea of these asexual life histories potentially being correlated with relaxed and slowed molecular evolution. And so what I'm thinking about specifically here is this idea of sex bias and mutation rate, and specifically how for a lot of these asexual lineages, they are primarily unisexual and almost exclusively female, right? We'll discuss this in depth a little bit later on in the presentation, but I hypothesize that basically some of these strong biases in mutation rate that we observe throughout the rest of the tree of life might also carry over and correspond with relaxed rates of molecular evolution within our kind of unisexual female species specifically. So in addition to these three kind of compensatory mechanisms, something that I've taken an interest in recently is kind of leveraging modern methods to kind of increase our temporal resolution at which we're able to answer these questions. And so these modern methods I think about specifically are ancestral recombination graphs. And that's something we've heard quite a bit about this quarter already. But basically what these let us do is visualize and infer genealogies as we move across genomes, right? So instead of now inferring just a single phylogeny capturing relationship between individuals that is meant to represent an entire genome, we're able to look at windows specifically and see where those topological changes in these genealogies have occurred. They're a super, super powerful tool for letting us get temporal resolution to how often these kinds of gene conversion events or gene flow events are occurring. And we'll talk a little bit more about the process we've used to infer these for some of my specific chapters. So getting back to these three different mechanisms now, the way that I've kind of structured my dissertation is basically to have individual chapters kind of investigate each of these distinct mechanisms. So we have our first chapter kind of looking specifically at that process of integration, our second chapter looking at the process of mitotic recombination, and then our third chapter kind of looking at the pace of molecular evolution within an asexual lineage. And that asexual lineage being specifically the Amazon model. And so this is the context under which I'm investigating all three of these different questions. The Amazon model is a super, super cool system, and it's very, very unique in the sense that it is quite evolutionarily ancient in origin. And like all other kind of vertebrate asexual lineages, it has origin that's associated with hybridization. And so specifically, we hypothesize, and the evidence pretty strongly supports a hybridization event occurring approximately 100,000 years ago in the past, which translates to about 400,000 generations. And then all modern clonal gynogenetic Amazon mollies being descendants of that single hybridization event, and most likely a single F1 individual as well. So the mode of asexual reproduction that our Amazon mollies practice is what's known as gynogenesis. And this is a slightly modified form of asexual reproduction in the sense that these female Amazons are still producing diploid gametes. And so they're producing diploid eggs, but those diploid eggs require essentially pseudo-fertilization events in order to trigger the process of embryogenesis. The vast majority of the time, we'll see that the kind of sperm genomic contributions from those paternal sperm donors are not included into that kind of developing embryo. And so the result is still a kind of true clonal offspring, but which has experienced kind of close contact with sperm donor material in that kind of process of embryogenesis, essentially. And so there's a wide variety of different species which can serve as sperm donors for this gynogenetic lineage, but most of the time in wild kind of natural populations, we see one of the two parental species involved in that hybridization event kind of serving as sperm donors. The range for this species kind of is right at the intersection of the two paternal lineages, which we think are most closely related to those kind of initial hybridizing individuals. And here on this distribution map, you can see highlighted in blue the distribution of one of our parental species, highlighted in orange the distribution of the other parental species, and right in that range of overlap, you can see this distribution highlighted in red, which is the range of our hybrid kind of asexual lineage. So you can see pretty confined to this region of the central Gulf of Mexico, kind of straddling the northern border of Mexico and southern Texas, essentially. So a little bit about the evolutionary history of this lineage. Now, we have pretty strong evidence that, like I mentioned initially, all modern gynogenetic clonal Amazon mollies descend from a single hybridization event that occurred approximately 100,000 years ago. And this is supported by both our mitochondrial DNA, which was used to generate phylogeny in this case, as well as DNA from several nuclear microsatellite markers as well. So across both of these, you can see that there's strong support for these Amazon mollies forming a monophyletic clade. And where we've done the additional process of the phylogenetic dating, we can see that that kind of date corresponds to approximately 100,000 years ago as well. So something else that's kind of interesting about this lineage is that despite having persisted for those 400,000 generations, there's also no real evidence of that relaxation of purifying selection that we would expect given the absence of recombination. And so recent studies that have done this have mostly characterized our kind of polymorphism patterns across each of these species involved in the species complex. So again, Formosa representing our asexual lineage, and then Latipina and Mexicana representing our two sexual ancestors. And you can see that our patterns of polymorphism, so in this case, our polymorphisms that are non-synonymous as compared to our polymorphisms that are synonymous, remain fairly equal across our three kind of species involved in this complex, as compared to a kind of increase in the number of non-synonymous polymorphisms that we might expect in our hybrid asexual lineage, given that there has been some relaxation of that purifying selection. Additionally, we can also see that as we compare across variants that carry different kind of functional consequences, which is what's represented here on this y-axis, right? We have variants which are either low, moderate, or high in functional consequences. We can see that in our asexual lineages, represented by these blue bars, there has been a reduction in the number of these heterozygous variants which are high kind of in functional impact. Again, pointing towards the action of that purifying selection and maintaining genomic function within this lineage. So all of this to say, despite the absence of recombination, we really don't see any patterns of genomic evolution which suggest strong differences from closely related sexual species. So let's dive into our chapters a little bit. Our first thing we're going to talk about is that kind of process of introgression and how specifically that plays into this system. So we're going to title this Prevalence and Long-Term Consequences of Gene Flow Within an Asexual Lineage. So starting from a kind of cytogenetic and phenotypic perspective, remember that we have this kind of system where each generation or offspring are coming into relatively close contact with the sperm-doning species, not of the same species as the hybrid lineage, but typically one of the two parental species. And there is that kind of opportunity for interaction with the genomic material from that sperm-doning species, right? And so how evidence of introgression first kind of showed itself was in actually phenotypic data, where in this case, we have panel A here showing our typical pigmentation phenotype for our female Amazon mollies, right? Again, this is our hybrid clonal gynogenetic lineage. In panel B here, we have the pigmentation phenotype of a male sperm-doning species that we use for a certain number of kind of laboratory breeding events, essentially. In panel C and D, we can see these pigmentation phenotypes that are associated with offspring from the sperm, from the kind of fertilization events involving this pigmented male sperm donor species. And what you can pick up on is obviously there's been some transmission of that kind of pigmentation phenotype we typically associate with the sperm doning species into the offspring of those kind of events which use the sperm doning species as a sperm donor. When we look at things on a cytogenetic scale, we can also pick up on these extranumerary fragments that are associated with these kinds of sperm doning events, right? And so specifically, anything that's kind of highlighted in the box here is extranumerary or extra to the typical kind of 23 chromosomes that we'll see in our Amazon mollies. And so obviously there's some variation in the number of these different microchromosomes or extranumerary fragments that can be carried by individuals, but that is evidence for this kind of transmission of genomic material from sperm doning species into our hybrid lineage that is otherwise asexual. When we look at things from a genomic perspective, we can also pick up on signals that are consistent with these kinds of extranumerary fragments as well. So something that's kind of important to note here is that because the sperm doning species are usually pretty closely related to our hybrid lineage, we will oftentimes have those extranumerary fragments be fairly homologous to certain portions of the genome. And so what happens is when you do things like alignments, those extranumerary fragments will show up as variations in copy number as well as variations in allele depth as well. That kind of differs from what you would expect under a typical kind of diploid genome scenario. So specifically, when we look at copy number, which is represented by panels B and also to some extent panel C here, we can see that there are certain scaffolds or regions of the genome where we see certain individuals in this kind of population data set have this signal of increased copy number. Specifically, you can see in panel B, a pretty clear example of an individual which has copy number centered at 3 rather than at 2, indicative of an extra copy of that kind of scaffold being represented within the sequencing data. Additionally, when we look at the kind of depth of our two different alleles at a heterozygous site within the scaffold, right, you can also see that evidence for kind of this bimodal distribution of allele depth, which we would not expect under a true diploid genome kind of scenario, right? Obviously, when you're sequencing genomes, you expect to have approximately equal representation from each of the alleles at a locus. When that's not the case, that's usually indicative of more than one copy or more than 2 copies in this case of that locus being present within your sequencing data. So again, all these signals here are kind of consistent with what we would classify as an extranumerary fragment, the result of gene flow within this species. So despite all of these developments that have been made, there's still quite a few questions that remain regarding these kinds of gene flow events, right? Specifically, we're relatively uncertain of the kind of frequency that's associated with these paternal introgression events, and specifically, at the rates that have been previously identified, just based off of scanning genomes and seeing how often we pick up on these extranumerary fragments, the mechanism of gene flow itself at the rates previously identified isn't sufficient to kind of compensate for the extent of, or to push the age of the lineage to the extent that we see empirically, right? And so what we kind of predict is that if introgression in this case, or if gene flow is kind of serving as a compensatory mechanism, we should be able to detect higher rates at which these kinds of events are occurring, right? In addition to this, there's also been no attempt to put some kind of temporal scale on the frequency at which these events are occurring. So the extent of what's been done is, like I said, scanning genomes and seeing how often we pick up on these types of events, but no one's tried to estimate really a per generation rate at which we're experiencing these kinds of gene flow events in the incorporation of extranumerary fragments. Additionally, it's also unclear if extranumerary fragments are the only mechanism by which we can pick up on these or by which we can experience these types of gene flow events. So specifically, you oftentimes hear within the Amazon Molly literature, people talking about this concept of paternal leakage, sometimes as something being separate to the microchromosomes that have been detected elsewhere, right? And so if we do pick up on any evidence of these kind of gene flow events not associated with extranumerary fragments, we should be able to find them and associate them with kind of gene flow, but not associate them with the typical microchromosome signals that we would expect, like the increased copy number and like the bias in allele depth as well. So to kind of address some of these questions, we're applying a couple of different methods to some existing data sets. So first of all, we have a data set of 8 hybrid individuals, which we've sequenced from within our kind of lab lineages, right, which we're taking advantage of to kind of basically recapitulate and repeat this identification of microchromosome kind of analysis using next generation sequencing data with the specific goal of trying to identify these shared microchromosomes, which should provide evidence for that kind of long-term inheritance, which is necessary for kind of compensation for mutation accumulation. Additionally, there's also a data set of 19 hybrid individuals, which are sequenced from these wild populations, which we're also using to search and scan for this evidence of gene flow that's not associated with the microchromosomes. And we're applying some new methods there that let us move past the typical kind of microchromosome signatures that people will use to pick up on gene flow. So for our identification of paternal introgression events, we're once again relying on these 2 specific signals of our copy number variation, and in addition to that kind of allele depth bias as well. And we're separating these putative introgression events from other forms of copy number variants by additionally kind of calling copy number variants using other software which relies on different metrics other than copy number variation specifically, and then kind of intersecting those identified copy number variants with our putative kind of microchromosome gene flow events to remove the set of kind of gene flow events which actually probably correspond with copy number variants derived from other kind of mechanisms. Additionally, we're also incorporating some of those ancestral recombination graph kind of methods as well, right? In the sense that we're going to be, first of all, using some modern kind of software to infer these ancestral recombination graphs, and then doing some fairly thorough kind of validation techniques to make sure that our genealogies are actually capturing what the true relationships between our samples across those genomes are. We're then going to be scanning our genealogies for these coalescence events, which are consistent with gene flow, specifically where we see kind of hybrid haplotypes reconstructed as being external to the hybrid clade specifically. And finally, we're going to be mapping these identified microchromosomes onto our ancestral recombination graphs to get that kind of temporal scale, right? Specifically to estimate the frequency at which these gene flow events are occurring. So, talking a little bit about our results now, the first thing we can see is that when looking across our lab sequence of individuals, remember this is that first data set, which includes 8 individuals from our lab populations, we can see that there's a strong kind of signal of copy number that we pick up on at several different locations throughout the genome, right? Specifically in this case, we're looking at a kind of increase in copy number that's shared across 4 different individuals, all kind of belonging to the same formal lineage, and which also spans about 170,000 base pairs within that genomic window. So this is for a specific chromosome, obviously, and it's also supported by this kind of bias in allele depth as well. As you can see in this plot on the right, rather than having our reference allele depth centered around 0.5, which we would expect under equal representation of each of those alleles, we instead have that kind of reference allele relative depth for these specific samples, which have the copy number increase centered at around 0.66, which is, again, 2/3 or what we would expect under a scenario of having 3 copies present at a locus rather than 2. So what we've now kind of switched to doing is trying to use some of those ancestral recombination graphs to, again, add temporal scale to the frequency at which we're kind of defining these events. So the first thing we'll kind of pick up on is that in this kind of array that we've reconstructed, which captures a specific window of the genome again, right? You can see that there's 2 major clades which we have identified within this array. Our tips here are colored based off of the sample, based off of the species that they're associated with that kind of legend up in the left, identifying our red color tips as those belong to our hybrid asexual lineage, red P formosa, and our blue and our green tips representing our 2 other kind of parental sexual lineages. So the fact that we can reconstruct these 2 lineages in this way is good news because it allows us to identify that there are, in fact, 2 distinct kind of lineages which group with the two distinct haplotype groups within these hybrid individuals, which group with their specific kind of parental species. Looking closer at one of these haplotype groups, we can also see that there's kind of a clade of hybrid haplotypes that's kind of separate from our parental sexual haplotypes. And in addition to that, also some of our hybrid haplotypes, which have grouped with our kind of parental haplotypes rather than with the typical hybrid haplotypes. And so going back to those methods, remember we were talking about these patterns of coalescence, which we are not consistent with like complete isolation from the parental species, right? And so in this case, this kind of shows that signal where we have these unexpected coalescence events associated with these hybrid haplotypes, basically being more closely related to some of the sexual haplotypes as compared to what the other hybrid haplotypes, which isn't really something we would expect to see in the absence of gene flow. So signals like this is exactly what we're looking for when we're trying to pick up on gene flow outside of these kind of microchromosome events, right? And again, this is not informed by any kind of copy number variation. It's strictly based off of relationships between the haplotypes. There have also been quite a few roadblocks that we've experienced with this kind of hard inference process in the sense that our expectation going into this is that across the chromosome, we should expect to see our hybrid haplotypes consistently associate with specific parent ancestry, right? When we actually look in the empirical data to see if that's true, what we instead see is that across each of our hybrid haplotypes, represented by one of these bars here, there are 38 bars that represent 38 haplotypes across 19 different individuals. You can see that there's approximately equal representation from each of the parental species, represented by the different colors for each of these parental haplotypes. this is like the opposite of what we want to see, and this does not give us good kind of confidence in the kind of genealogies that we've inferred, right? And so what this is preventing us from doing is making inference across entire genealogies rather than looking at a single genealogy specifically. So what we would like to see is that for each of these bars, that bar is primarily yellow or primarily purple, right? Representing consistent association with one specific parental ancestry. Instead, you can see that it's a vast mixed match across the genome as we look across this chromosome specifically. On a more finer scale, we can see that there's even like the absence of distinct haplotype blocks specifically, right? So this is for a specific sample comparing one of the haplotypes above to the other haplotype below. And you can see that we switch phases across our haplotypes on a very, very rapid scale. There's not the extended kind of haplotype blocks, which we would expect under the absence of recombination, right? And so if recombination were acting in this system, this wouldn't be a concern because this is exactly the kind of signal we would expect maybe. But because we don't have recombination acting here, we expect to see those consistent associations with parental ancestry. That's just not something that we picked up. So a little bit of further investigation into this has identified the issue as probably being the phasing process of our genotypes prior to doing the ancestral recombination graph inference, right? And so basically the phasing of these hybrid non-recombining genomes is proving to be a lot harder than we thought it would initially be. We've arrived at what we think is potentially a solution where after kind of combining multiple different rounds of phasing using multiple different approaches and multiple different sample subsets as well, we're able to achieve a lot better kind of resolution and generate those kind of pre-phased genotypes prior to doing the ancestral recombination graph inference. So all of this is still kind of a work in progress, but we're hoping to have some results from these analyses pretty soon, now that we've resolved some of the initial issues we've run into. But yeah. So that was our first chapter, kind of looking at this mechanism of introduction, right? Let's talk a little bit about that second chapter now, this idea of mitotic recombination. And so much like our first chapter, this will be looking at the prevalence and long-term consequences of now these gene conversion events, again, acting within an asexual lineage specifically. So a little bit of history about the kind of identification and detection of these gene conversion events within the Amazon Wally system. This was first kind of described in a paper from 2018, where the rate of gene conversion per site per generation was estimated to be about 3.32 E-8. So again, this is a rate that's estimated very, very roughly simply by dividing the number of observed gene conversion events by the approximated age of the system. And so this does not involve any mapping onto phylogenies or any formal inference techniques. Again, a very, very rough kind of estimation, right? In addition to this, something else that the authors of this study kind of pick up on is that there's also no relationship between the distribution of these gene conversion events and the amount of divergence between the two haplotypes of the hybrid genome. This is illustrated well by this plot here on the right, where basically you can see across these different FST values as a metric of divergence, we see that there's no strong relationship with the number of gene conversion events that they pick up within these windows. More recent work has kind of identified these gene conversions potentially occurring at a much higher rate than initially thought. This was done through kind of reanalysis of data from previous studies and generates plots that look a little bit like this, where we see across a couple of different samples now, and within this specific kind of chromosome window, we see that there's a vast number of these kind of gene conversion events, some of which are shared across samples, other of which are restricted to certain samples. But this would imply a rate that's much higher than the 3.3 E-8 that was documented in the past. Arguably more interestingly, there's also an argument that's been made for the gene conversion events having a compensatory role as well. And the main evidence that's kind of used to support this is that there appears to be a strong bias towards converting away from private variation that we kind of that's kind of restricted to our hybrid lineage, right? So specifically variants that have arisen in isolation within our asexual lineage are more likely to be lost as a result of gene conversion events, right? So the paradox that kind of arises from this is that if gene conversion is as common as it seems, and also if gene conversion is serving this kind of compensatory role and it's being argued by this study, then why do we see the fixed F1 haplotype structure within our species, right? Why do we see that there's no evidence of recombination at all? Because sufficient levels of gene conversion should show up as like recombination events in the sense of being able to restructure those haplotypes. This brings us to kind of a couple of the questions that we're attempting to address with this chapter. First of all, trying to get a better sense of what that per generation frequency of gene conversion is. So looking at this question, trying to understand if gene conversion events occur at sufficient rate to kind of compensate for the mutation accumulation that we would expect given the age of the lineage. And additionally, how gene conversion events relate to our putative gene flow events that we kind of investigated and talked about in chapter 1. Specifically, our canonical mechanisms of gene conversion are oftentimes kind of associated with very, very specific signals in terms of track lengths being relatively short, right? And so we're looking for evidence of these longer gene conversion tracks, which don't align well with the canonical means by which gene conversion events kind of occur. So our methods we're kind of looking into this are, again, making use of those same 2 existing data sets where we have 8 hybrid individuals from our lab lineages sequenced to relatively high coverage, in addition to these 19 hybrid individuals that are from our wild populations, right? Again, all existing data and stuff that we didn't necessarily generate for this study. In terms of identifying the loss of heterozygosity events, our first approach is going to be to look specifically at sets of kind of fixed differences that we identify from our parental species, and then look for evidence of these contiguous fixed differences which have lost heterozygosity, right? Which we're going to call our loss of heterozygosity tracts or loss of heterozygosity regions. So that cutoff specifically is, again, 3 or more of these regions have to be, 3 or more of these fixed differences have to be contiguously lost in order to kind of be classified as a loss of heterozygosity region. In addition, we're going to try to move beyond these fixed differences and also scan across our genealogies, again, taking advantage of those ancestral recombination graphs to look for these topology changes, which we think are consistent with gene conversion. So we'll talk a little bit about more of those in just a second. Once we've actually identified our gene conversion events, our idea is basically to quantify these rates of gene conversions by mapping these events onto our ancestral recombination graphs, which are obviously calibrated by time, right? And we're then going to extrapolate across these posteriors of tree sequences to generate a posterior distribution of what we think that rate of gene conversion should be. And additionally, we're going to look at specifically these long loss of heterozygosity events, if we pick up on any, those that are inconsistent with our canonical methods of gene conversion, or mechanisms of gene conversion. And within these long loss of heterozygosity regions, when I quantify the degree of polymorphism sharing that we see with parental species of whose ancestry is basically retained at the locus right there, with the expectation being that within our long loss of heterozygosity regions that are derived from gene flow events, we should see increased sharing of polymorphism with parental species, given that those long loss of heterozygosity regions are derived from gene flow events. So a little bit about our results now. Kind of a sanity check we did before any of our further gene conversion analyses is basically to see if our patterns of linkage disequilibrium are consistent with kind of haplotype restructuring by means of gene conversion. And what we see is that for the most part, that's generally true. We look across kind of a specific, comparing across loci that are increasingly distant in this case, right? From anywhere right next to the kind of focal locus, all the way up to the megabase away from the focal locus. And what I kind of want to draw your attention to is that within specifically the first kind of 100,000 base pairs or so, the major change that we see in linkage disequilibrium is that within that first window, there's kind of a massive drop in LD as we move farther and farther away from our focal locus. And so what this basically shows is that these mechanisms of genome restructuring are happening mostly on small scales as we move farther away from a locus, more than about a kilobase or so in this case. We start to see that LD decay slightly. But once we move beyond that 1 kb kind of window, there's no longer any decay beyond that. So anywhere in this region from like 0 to 250,000 base pairs away from our focal locus, we don't see any strong patterns in that LD. Where we would see those strong patterns of LD decay as we move kind of consistently farther away from the locus, if we're talking about recombination kind of structuring these haplotypes, right? So specifically, the fact that we're seeing mostly restructuring within that short kind of 1 kb window tells us that most likely what's going on here is that we have restructuring primarily through the means of these gene conversion events occurring on small scales within the genome. Additionally, we've also gone through and identified these loss of heterozygosity regions, and we see that they're widespread in both our wild populations, right? You can see colored above our chromosomes here are numbers of individuals that are sharing the loss of heterozygosity event with kind of darker colors representing, with lighter colors representing more individuals sharing the event. And below our chromosomes, the ancestry that's kind of retained across the lesion, right? So either one of the two parents or a mix of ancestry across different individuals. Same kind of patterns can be observed when we scan our lab populations as well, where we see evidence of those strong loss of heterozygosity events. Looking across the genome, we can see that this is not something that's restricted to certain chromosomes, and that is fairly consistent genome-wide, right? We see evidence of these loss of heterozygosity events, and interestingly, some that are also fairly long in length. So, for example, we can look at chromosome 18 here, where at the end of that chromosome, we have a fairly long loss of heterozygosity region, perhaps spanning multiple megabases or so, which consistently has lost heterozygosity in that same direction, right? When we look at these long loss of heterozygosity regions specifically, something that we don't pick up on is any evidence for that increased polymorphism sharing, right? And so, like our plot before, we're looking at the number of individuals that are sharing loss of heterozygosity region being plotted above our chromosomes, and then now below our chromosomes, we're looking at the amount of shared polymorphism that we see within the specific windows. So by amount of shared polymorphism, I'm basically referring to triallelic sites in our parental species that also appear to be triallelic within the hybrids as well. So within these long regions, like this one on chromosome 6, for example, we don't really see any evidence of that increase in shared polymorphism. What that's telling us is that these long loss of heterozygosity regions are most likely not originating from gene pool events, but from kind of within haplotype restructuring, right? There are a couple of these mechanisms which can generate longer tracks of loss of heterozygosity and longer gene conversion events, but that haven't been described super well in most systems. So I'm thinking things like specifically break-induced replication is something that we know can happen, but that just hasn't been really well documented outside of, in our case, supposedly with these kind of long loss of heterozygosity tracks within this asexual non-recombining system. So similarly, we can also look back to our ancestral recombination graphs as well for evidence of these kind of gene conversion events. And specifically, what I'm going to point your attention to here now is the number of hybrid haplotypes that falls within each of these kind of parental clades. So again, we have two large parental clades in this ancestral recombination graph, right, in this genealogy. And in our first kind of parental clade, this is our P latipinoclade, one of those parents. And we observe that falling within this clade is kind of 21 separate hybrid haplotypes. And falling within the other parental clade is 17 of these other hybrid haplotypes. Under a scenario of no recombination, what we would expect is that there's approximately equal numbers of hybrid haplotypes falling into each of these parental association clades. But what we see is kind of the opposite of that, where there is this kind of imbalance of hybrid haplotypes associated with each parent. Again, this kind of signal is what we would expect given gene conversion, but the ability for us to kind of extrapolate and infer things on a larger scale outside of single genealogies is kind of limited by the kind of lack of confidence that we have in the current set of genealogies that we've inferred using these techniques. So that brings us to the end of our second chapter, kind of looking at that process of mitotic recombination. We're going to wrap up by talking super quickly about this pace of molecular evolution stuff and what exactly is going on here. So we're going to title this Evolution of our de novo mutation rate within an asexual lineage. I want to start off by talking a little bit about this sex bias stuff that we think is going on, right? So specifically where this originates from is when you have two sexes in the species, oftentimes you have separate gametogenesis pathways as well that are generating either eggs or sperm most of the time, right? Our oogenesis pathways are oftentimes going to involve a much lesser number of cell divisions as compared to our spermatogenesis pathways. That lesser number of cell divisions basically means that our oogenesis pathways have less of an opportunity for these mutations to occur within the germline as compared to our spermatogenesis pathways. What this results in is that across the tree of life, this is across a broad range of taxa here, we see that there is strong bias in where mutations kind of originate, in the sense that a lot of our mutations are coming from male gametogenesis pathways as compared to female gametogenesis pathways, right? So we'll typically quantify this using what's called an alpha parameter, comparing the rates of mutation in our male versus our female kind of gametogenesis pathways. And you can see that across all these taxa, that alpha parameter is for the most part a lot greater than 1, with those kind of confidence intervals oftentimes not even overlapping 1 at all. So there is a strong bias in kind of in mutation rate that's associated with sex that's well documented across all of these different taxa. Specifically, if we look at close relatives of our asexual Amazon mollies, we see within the common guppy, for example, that the male to female mutation rate ratio, that alpha parameter, is about 1.5 times. And so what that means is that there's 1.5 times more mutations that are originating from male gametogenesis pathways as compared to female ones. Additionally, there's also some other interesting signals that have been picked up on by some more recent studies, which suggest perhaps haplotype-specific mutation rates, something which hasn't really been super well documented in other systems. What we're looking at here is on the x-axis, the divergence between one of our kind of hybrid haplotypes and the most recent common ancestor of all of those hybrid haplotypes, as compared to divergence on the y-axis between the other hybrid haplotype and the most recent common ancestor of that other hybrid haplotype. Consistently, what we see across all the samples is that there is a higher divergence that's associated with one of those haplotypes as compared to the other, right? We consistently see that samples are falling above this kind of 1-to-1 ratio line where we would expect if divergence rates were approximately equal across the two haplotypes, suggesting again that these haplotypes might have specific mutation rates that are divergent essentially. So the kind of question that we're going into this with is how germline mutation rates in an asexually reproducing lineage compare to those that are from a sexually reproducing close relative, basically. And our hypothesis is fairly simple. Given the absence of kind of mutation-generating gametogenesis pathways, those that are generating sperm, right? We expect that the Amazon molly will experience germline mutations at a pace that is slowed relative to or related sexual lineages. So our methods to kind of go through this are basically to do a kind of novel breeding experiment, a parent offspring sequencing approach, essentially, where we're going to estimate mutation rate for both our Amazon mollies as well as one of those parental ancestors. We're going to replicate across multiple different clonal families as well as separate families, and then also sequence multiple offspring per family as well. We're going to sequence parents and offspring from each of these families to about 30x coverage using a variety of different sequencing technologies, and then do stringent kind of filtering processes to identify the number of de novo mutation rates, de novo mutations that we can observe in each of these distinct families to calculate that mutation rate, and then do statistical comparisons using a kind of Wilcox and Simon branch test. So our predictions for this is, again, that in the absence of our spermatogenesis pathways, we see reduced mutation rates as compared to our sexual lineages. And this should kind of hold true across the multiple different breeding families that we're using for this study. And so in a graphical format, this is kind of what we expect that to look like, where we have our purple kind of samples from our families here representing our asexual lineage, and then our yellow samples representing our sexual lineage. And you can see that there is a significant difference between these two different kind of simulated data sets, right? And this is what we would expect given the difference in mutation rates that is aligning with that kind of alpha parameter that we expect from closely related species. So to kind of review super quickly, we have our first chapter where we're looking at the process of introgression, where we've identified these putative gene flow events which appear to be heritable, right? And then we're going to expand on that by doing inference essentially with these ancestral recombination graphs to add that temporal scale and estimate the rate at which these events are occurring. We've got our second chapter looking at our gene conversion events, again, which we've identified being pretty rampant and common throughout multiple different data sets. And again, we're going to make use of and take advantage of these ancestral recombination graphs to, again, estimate frequency and add some sense of temporal scale to these distinct gene conversion events. And finally, we've got our third chapter kind of investigating that pace of molecular evolution. So looks like I wrapped up right on time, but some quick acknowledgements. Obviously, I've had a lot of excellent mentorship during my time here at Davis. I've now associated with 3 separate labs, all of which have been absolutely amazing in terms of guiding me through this process and helping me to carry this stuff to fruition. So Kate, obviously a wonderful mentor, Andrew and Graham as well have been excellent sources of expertise. Each of these labs, I've spent significant time with as well and have contributed to both making my time here at Davis more enjoyable and also keeping me sane in all these different things that I've been doing. I would like to thank my QB committee who provided excellent feedback during that process I was going through and has contributed a lot to kind of shaping the way that my dissertation was played out. I'd like to thank my funding sources, obviously. I was the recipient of a graduate research mentorship fellowship during my second year, which helped to ease the process of doing that QB significantly. I've also received CPB funds in the form of the Daphne and Ted Pinkley Award in Evolutionary Biology for 2 consecutive years now. And a lot of my computational resources have come in the form of an NSF Access project, which I applied for, which obviously, in the absence of a lot of this computational work, would not be possible. And finally, I'd like to thank the amazing population biology graduate group, which has been a huge part of my time here at Davis and which I don't think I would have been able to proceed so far in my dissertation without. But yeah, thank you everybody for listening and happy to take any and all questions. Yeah, Carolina.
Wow. Crazy. Yeah, I'm really proud of you, Max. I was wondering if you had any thoughts on how like epigenetic modifications might impact this, considering they're asexual lineages.
Certainly, yes. So there are other people in the lab who I think would be able to speak a lot better to this. But one of the primary mechanisms which we're seeing change on a generation to generation basis is likely through the action of different epigenetic kind of mechanisms, right? So we have things like methylation, which we know for sure are probably going on within the system that are resulting in these individuals actually differentiating a fair bit in phenotype, even if their genomes are kind of remaining the same. Yeah, follow up.
I have one more question. So I just learned that there are some seagrass populations that have not flowered. And so I'm wondering if there are like, do they do this in plants? Like, have you been like plants? Or is this primarily looking at animals primarily? Or what do we know? Yeah,
Sure.
about other systems with this stuff.
Yeah. So I will say that for plants, a lot of these alternative reproductive modes are a lot better documented than for these kind of vertebrate lineages, right?
Okay.
Although interestingly, I'm not sure if I'm familiar with any plant systems which actually undergo gynogenesis. That seems to be something that's restricted to these higher order vertebrates. But what you do see in a lot of plants is tons and tons of self, obviously, right? So that is something that's like pseudo-asexual in the sense that individuals are able to reproduce by themselves, but on a mechanistic basis, it actually differs quite a bit in the sense that you still have recombination acting and are perhaps able to avoid some of those kind of genomic consequences. I'm sure there's tons and tons of other weird kind of plant reproductive methods that I'm just not familiar with, but yes, lots and lots of cool stuff going on for sure. Any questions? Yeah, continue.
Great talk. This is amazing. And I probably have a lot more questions for thinking about your mitotic recombination and the larger regions of homozygosity that you observed. I noticed that they were primarily at the beginning and ends of chromosomes, and I was wondering if there's any particular reason as to why they're more commonly found there.
Yeah. So there's a couple of things that might be going on with that. One thing that might be going on is that part of this could be action or concentration of these types of events, specifically in our kind of gene conversion regions, right? Another thing that might be going on is that I mentioned super briefly this idea of like break-induced replication. I am honestly not super familiar with the mechanisms of that specific kind of repair mechanism, but usually how that works is that after experiencing a double-strand break, you'll basically join with that kind of double strand break the entirety of the rest of the chromosome from where that kind of double strand break occurred. And so something that I personally hypothesize might be going on there is that we see these starts of these kinds of loss of heterozygosity regions being where we have, for example, double-stranded break having occurred, and then the rest of that being the result of a break-induced replication event kind of generating the loss of heterozygosity signal. Any other questions? Hey.
The fact that the mutation rates tend to be higher in male gametogenesis than female, I thought that was really.
Yeah, it's super
Yeah.
cool. Yes.
Do you, could you briefly like go over are there ideas about why that exists or?
Yeah.
whatever?
So the main hypothesis we have about why that exists is because of the different pathways associated with male versus female gametogenesis. And so, I'm actually thinking of the. Here we go. So one of the main differences between our spermatogenesis and our oogenesis pathways is that our oogenesis pathway is typically involving a limited number of these kind of cell division events, right? You're generating your oocytes fairly early in your kind of life history, and then you're not really doing a lot further of these kind of cell division events. As compared to our spermatogenesis pathways, which are almost continuously dividing to generate more sperm, essentially, right? Each one of those divisions is an opportunity for mutation to occur within the germline. And so because of the fact that we're having more kind of cell divisions within the spermatogenesis pathways, that is, most people think, why we're experiencing more of these germline mutations within our spermatogenesis pathways as compared to our oogenesis.
Got it, thank you.
Questions?
Oh, thank you so
Awesome.
much.
Thank you guys so much.